Assalamu alaikum. Good afternoon, everybody. It is my pleasure to welcome our visiting professor, Professor Henry J. Klassen, who is professor and director of Stem Cell and Retinal Degeneration Program at the Gavin Herbert Eye Institute, Department of Ophthalmology, University of California at Irvine, and also adjunct scientist at the Skippins Eye Research Institute, Department of Ophthalmology at Harvard Medical School. Professor Klassen holds a PhD in neurobiology, anatomy, and cell science from the University of Pittsburgh and a medical degree from the University of Pittsburgh. He also did his residency in ophthalmology at the Yale University and a fellowship in retina and in medical retina and retinal research in Moorfields Eye Hospital and the Institute of Ophthalmology in London. Professor Klassen's career focus has been on cellular therapy for the dysfunctional retinal pigment epithelium and retina, including cell therapy delivery methods, regenerative medicine, and translating science into cell and gene medicines. He's had extensive research in the domain of stem cell transplantations and protection of the outer retina in pathologies such as retinitis pigmentosa, but also age-related macular degeneration, including 3 completed phase 1 and phase 2 clinical trials on the safety of intravitreal injections of human retinal progenitor cells, called J cells, in adult subjects with retinitis pigmentosa. He's had an extensive CV, which I had to review yesterday, which includes 12 awards, numerous invited presentations, authorship of about 90 journal articles and 19 book chapters, most of them practically pertaining to stem cells, retinal transplantation, and tissue engineering. And he holds 8 patents related to isolation and transplantation of retinal stem cells, compositions and methods for treating retinal disease, and potential methods of human retinal progenitor cells isolation and culture. He's a co-founder of a company called JSite, which we will be hearing about today, and his talk today is indeed about the retinal progenitor cells therapy in the treatment of retinitis pigmentosa, something that we're very excited about. So please welcome Professor Klassen. Thank you.
Thank you very much for the kind introduction, and thank you for the opportunity to present our research today. This will, by necessity, be a brief overview, but hopefully it will whet your interest in what we're doing here with retinitis pigmentosa. I also direct you to my disclosure statement. As you heard, I do have a company founded to commercialize this technology. Okay. Is this working? Yes. Okay. Good. So you just heard about retinitis pigmentosa, and I don't want to belabor the issue, but one of the points I want to make is, although this is a rare disease, there are many underlying genes that can cause this. So it's a final common phenotype for a whole host of different genetic conditions. And that poses certain challenges from a potential treatment perspective. But of course, the main takeaway right now is that this is a very difficult condition to treat, which is why I've been working on this for so long. Here you see some images of the histology of the retina, and you can see that what starts out as a nice normal-looking retina and is able to provide for the patient nice normal vision will slowly degenerate to the point where you lose virtually all photoreceptors if you live long enough. And there's potential ways we could go about trying to treat a condition like this. And one of the most popular is gene therapy, particularly because this is a genetic condition. But as I just mentioned, the genetic heterogeneity of this condition makes individualized gene treatments somewhat challenging in a rare disease, particularly. Another thing to...
We have another mic.
Another thing that makes it challenging is that because the rods are expressing the mutant genes, and the rods are the first cells to be degenerating, then as the disease progresses to the point where the patients need treatment, it could be the case that there are relatively few rods left in the retina for your gene therapy to act upon. Another potential way to go about this is using a cell therapy as a means of cell replacement. In that case, you could replace cells. The challenge there is what you really need are cones at the late stages of the disease, and scientifically it's still a challenge to generate a high purity of cones in the laboratory that you could transplant. So that leads us to a third possibility of neuroprotection. There's more than one way you could go about that, but the first thing I want to do is provide the rationale for neuroprotection in RP. One of the first things to point out is that a neuroprotective approach would be gene agnostic, so you would not be treating each individual genetic subtype of RP. It would just be RP in general. And again, as I mentioned, the rods express the mutant gene. They degenerate first. But when the rods are lost, from the patient's standpoint, that just creates an impairment in night vision, which is different than going blind. But unfortunately, there's this knock-on effect where the cones eventually die. And the interesting part about this is the cones are not expressing the mutant gene. So they're lost later in the disease, and that imparts a huge devastating problem for the visual capacity of the patient. But interestingly, the loss of cone photoreceptors is not due to the genetics directly, it's due to metabolic imbalances due to the loss of the rods earlier in the disease. So neuroprotection basically can reset the metabolism of the cones, and when this is done, it can make a huge impact on the visual experience for the patient. Now, one potential way to mediate your neuroprotection is by way of a stem cell. So first I want to introduce what we're talking about in terms of stem cells. So what we have here is a kind of wandering pathway for development, human embryonic development. Starting with a fertilized ovum, you progress to a cluster of cells and then to a blastocyst. The blastocyst will implant in the uterine lining. A placenta will be generated, followed by a fetus. As this developmental process is occurring, the cells within the embryo are changing their capacity. And so different types of stem cells can be generated using donations taken from different stages of development. So if you take cells from the inner cell mass of the blastocyst, and you would obtain these from, say, a donation from an in vitro fertilization clinic, then those cells from that inner cell mass can be grown in the laboratory, and these are known as embryonic stem cells. These cells are inherently immortal. You can grow them forever, and they're also known as true stem cells. And the designation, the technical designation, is a pluripotent stem cell, and that means they can make any cell type in the body. And I want to contrast that to cells taken at the fetal stage, where the tissues and organs are starting to differentiate, laying down the basic body structure of the developing fetus. Cells within those developing tissues may not be mature yet, but they're already restricted to their tissue of origin. So if you take blood from a fetus, you know, the stem cells within that blood will be restricted to generating blood cells. And similarly, cells where the muscles are going to form will be the stem cells generating the muscle cells. So we're taking the tissue from the developing retina. It's not a true retina yet, but when we grow those cells in the dish, they basically are the retinal progenitor cells, and that's our cell of interest. And over in the right-hand column, you see illustrations of these different cell types. a colony of embryonic stem cells up top, and down below you see retinal progenitor cells growing in the dish, and that's what they look like. And you can see they look quite different. Early work in the mouse showed that we can grow these cells in the dish and analyze and manipulate them that way. Starting with the green fluorescent transgenic mouse, we can grow cells that are going to also grow and form fluorescent stem cells, in this case the retinal progenitors, and that will allow us to track these cells following transplantation. And here's an example of what I'm talking about. In the right-hand frame, you see some green profiles, and those are the retinal progenitor cells after transplantation. In this case, these cells have taken up position within the retina of the mouse. But the part I want to make clear to you is the red signal, and that's the photoreceptor layer in the recipient animal. So these are photoreceptors that are dying off in these animals because these are dystrophic animals. And so in the far right frame, you see that the red layer is very thin, and that's due to loss of photoreceptors. Compare that to the frame on the left, and you see that there's preservation of photoreceptors. All I want to illustrate here is that the presence of the green cells is also having a treatment effect on the red cells. There's more of them. So that's what we mean by neurotrophic effect, preservation of photoreceptors due to your treatment. The other thing I want to point out is these animals were not immunosuppressed, and yet somehow these cells are surviving. Why is that? Well, we did a lot of work looking into why retinal progenitor cells are tolerated following transplantation and come up with a number of remarkable conclusions. So one is, I think as ophthalmologists, you know that corneas can be transplanted without immunosuppressing the patient in the majority of cases, and that's due to the fact that the anterior chamber exhibits a property known as immune privilege. Well, we also see similar types of things in the retina. So the retina appears to be an immune privileged site. So if you put cells in the retina, they're better tolerated than in other parts of the body. That may not extend to the RPE, though, so just keep that in mind. Now, an interesting fact is that the retinal progenitor cells also exhibit decreased immunogenicity. So it's not just that you're putting them in an immune privileged site, it's that the cells themselves are less likely to be rejected. And we know that because when we put them in a place that's not immune privileged, like the kidney capsule, that these cells still survive and are not rejected, even though they're not protected in that location. So that's fascinating. These cells are actually doing something that protects them from the immune system. And here's a really critical feature in terms of clinical significance, and that is that if we repeat the dosing, we still do not enhance the, foster a rejection process. So that's going to be really important when we think about this clinically. So we've done studies in mice where we've injected up to 3 times, injecting both the original eye, the fellow eye, and then a repeat dose in the original eye, and still the grafts are not rejected. Now, those results in mice have been extended to other species, including rats, cats, and pigs. And here's some work we did in collaboration with other laboratories. On the top row, you see we took cells from a green fluorescent pig. These are transgenic animals, obviously. And then we can transplant them to the subretinal space, as you see here, or to the vitreous cavity, as you see in the far right. And from there, the cells will survive, and then they'll slowly differentiate and have whatever treatment effect. Down below, you see an example of some of the work in cats. In Korea, they generated a red fluorescent transgenic cat, we could isolate the red fluorescent cells from the retinas of developing cat fetus and then transplant that into cats that go blind because of retinal degeneration. In this case, we're talking about the CEP290 mutation in the Abyssinian cat, a naturally occurring RP model. Then turning to the human cells, of course, this is the basis for considering a therapy, is to isolate these cells. I initiated this project at Children's Hospital of Orange County quite a few years ago. At this point, we know quite a lot about these cells. We know these cells are not true stem cells. They are progenitors because they're not immortal. They will divide in culture. We can amplify our supply of these cells, but eventually we have to go back to donor tissue. They're also fate restricted. They can make different cell types in the retina, but you're never going to see these cells make blood or liver or pancreas. And then meanwhile, these cells can be tested in animals, but we're not going to be able to learn much about the immunology in animals because when you transplant retinal progenitor cells as xenografts, they can often be rejected. And in fact, if you put human RPCs into mice or into rats, you can expect them to be rejected. And in those cases, we do have to immunosuppress the animals in order to get the results we're looking for. But that leaves the question of, can we put a human retinal progenitor into another human and do it without immune suppression? And so for that, we need the clinical trials. One final point before we get to the clinical trials is that although we can inject these anywhere we want, including under the retina, we can also inject these into the vitreous. And as you know, being clinicians, that's a very streamlined way of delivering these cells because you deliver Lucentis and other VEGF inhibitors on a routine basis. And we can apply that same treatment technology to the delivery of these cells. And that's what we do. We use the same syringes and everything. Once these cells are injected into the eye, they form these clusters. So what you see here is not one retinal progenitor cell. It's a whole little cluster of these cells. But you can see it can just sit there living in the retina and exuding the neurotrophic factors that these cells naturally make. And those factors are going to treat the retina. Meanwhile, the cells of the graft are going to slowly differentiate. And then, after surviving for a year or longer, they're slowly going to die off. And they don't migrate into the retina like I showed you in some of the earlier mouse or cat work. In fact, they just stay in the vitreous where the physician can keep an eye on these transplants. And they do not become vascularized. So you just saw a talk about choroidal neovascularization, macular neovascularization. Well, that's not an issue that comes up based on these cells. And then when the cells have completely died off, they're cleared out by macrophages, and then you can re-inject. Here's the proof of concept, starting with the human retinal progenitors and then testing them in immunosuppressed retinal college, RCS rat, the Royal College of Surgeons, a very dignified rat. And so, again, we can inject these into the vitreous of the rat eye, and you've seen this image before, and from there they're going to deliver. Basically, what we're doing is using these cells as a trophic factor delivery system. And so then after a year, after the full treatment effect has been realized in the rat, we're going to harvest these retinas. And here in the middle, you see a hole mount. So you take the retina out and you flatten it onto a slide. And from there, you can use confocal microscopy to zoom in on the photoreceptor layer and see what's going on there. And so in the right column, you see a comparison between a control eye and a treated eye in an RCS rat. And these tissues have been labeled with rhodopsin in red and middle wavelength cone opsin in green, and the control, the untreated, is seen in the upper right, and you see it's relatively devoid of any profiles. That means the photoreceptors are basically gone. And then compare that to the treated retina at the same time point in the lower right, and you see that there's quite a few different profiles, both rods and cones, that are still present because of the treatment. So that's the demonstration of the anatomical demonstration of a neurotrophic effect from a human retinal progenitor cell. And then we want to know that those preserved photoreceptors are functional, and we've looked at it two different ways. One's with a optomotor response you see on top there, and you see an improved, if you look at the green histogram bar, that's the improved spatial frequency resolution of the animals that have been treated versus the other bars that are controls. And then below that, you see the electroretinogram, and each histogram bar here is the B wave amplitude averaged across the group of animals for each condition. And the two bars on the right are, again, the control groups. And then progressively moving to the left, you see ever-increasing doses of human retinal progenitor cells are generating a bigger ERG response. Again, consistent with the idea that these cells are helping the retina function better. Now I shift gears towards the clinical program. Just a very brief overview of our program. But as we started the trials, we generated human retinal cell product using a GMP-like method suitable for clinical application. So we end up with these frozen cryovials. Each one can dose a single patient, and you generate hundreds of those to begin your trial. And this is just a review of what I just said. These are human retinal progenitor cells that are derived from allogeneic human fetal tissue. So they're derived from tissue donations, not from embryonic stem cells. They're delivered by intravitreal injection, which is very convenient for performing a clinical trial. And these cells are going to supply neurotrophic factors. In other words, we're not using them for cell replacement. We're using them for neuroprotection. And those are going to benefit the cones, but they also benefit other cell types in the retina. So you'll hear more about that. And because it's a neurotrophic effect, again, this is agnostic to genotype. And then once the cells eventually die off, although they do persist for quite some time, we can re-inject these cells. And then there's still this lingering question I posed earlier. If we inject these cells without immune suppression, as we're going to do in the trials, what's going to happen there? Are they going to be rejected or not? And so here's the first trial, the first patient being dosed at RVA in Los Angeles. And the question is, what happened? And I'm happy to report there was no immune rejection in this first trial. Not only that, we saw some evidence of improved acuity in these patients, particularly with a larger dose, which is the one in the lighter blue. And you see improvement over baseline of around 6 letters, which is kind of impressive, because with a neurotrophic effect, you're mainly trying to limit the loss of vision, and yet we're seeing an actual improvement. So that was very intriguing. And then we did an extension trial where we dosed the fellow eye. So think about that. The patients had all been dosed in one eye, and now we're going to treat the other eye. Well, that raises the bar quite a bit in terms of the immunology, because the first injection could sensitize the patient, and then the second dose could create problems that the first dose didn't. So what happened then? Well, again, no immune problems. So we really are starting to feel confident at this point that these cells are going to be well tolerated immunologically, and we don't have to have these patients on chronic cyclosporin or other immunomodulators. And then we entered into the phase 2B trial Again, enrolling anybody with RP, irrespective of the genotype, they did have to be adult patients. We selected a range of visual acuities that was broad between 2080 and 2800. And the reason that was chosen was just for the fact that we needed to be able to detect a change in visual acuity because that's our primary endpoint. The other thing to point out is that we made sure they didn't have other eye diseases that could confuse the story when we get the readout during the trial. Then patients were randomized in one of three different groups, so 6 million cells, 3 million, or a sham control. And then the primary endpoint was, again, visual acuity at 12 months, change from baseline. And this was, as you heard, this is a relatively large study for RP with 83 subjects enrolled and 80 actually completed the whole trial. Safety-wise, the cell's well tolerated, and yet again, with this larger group, we didn't see any immune issues. And one of the nice things was that the higher dose didn't have more complications than the lower dose. So here's our efficacy readout. With the higher dose, we saw this nice evidence of a signal with around 7 letters improvement in visual acuity, similar to the earlier trial, and reaching a maximum somewhere around 6 months that's more or less maintained to 12 months. And that's very much what we were looking for. What we were not looking for was this apparent improvement of 2 to 3 letters in the control group. So that variability there undercut our statistical analysis to the point where we just missed statistical significance. So because of that, we said, well, is there something we can do about this variability in the control group? And so something we've seen is in the scientific literature, if you have a big difference between the two eyes when you start the trial, a large disparity in acuity, and you treat the worst seeing eye, that eye can spontaneously improve, even if it's in the sham group. So that accounts for why these patients can get an improvement, and that can affect the variability in the sham. And of course, we had to treat the worst seeing eye because this is a clinical trial and the safety is not yet assured. So the FDA wants you working on those poor seeing eyes. So taking that into account, we went back to our data and we said, we'll exclude all patients who had more than 15 letters difference between the two eyes. And when we did that, sure enough, the sham effect was lower, and we saw statistical significance at both 6 months and 12 months. Again, that's post hoc, but it showed that our analysis was correct, that going forward, if we restrict our entry criteria, we can have a positive impact on outcome. Going further, we said, well, since this is neuroprotective, you can't protect photoreceptors unless there's still photoreceptors around. In other words, if all the photoreceptors are gone, we can't have a treatment effect with a neuroprotective strategy. So as a surrogate way to kind of get a handle on how many photoreceptors a patient has going into the trial, we looked at visual fields, and we came up with this number of a central visual field of around 8 degrees is probably a minimum number in terms of how much preservation you want in your patient population to get optimal response from this particular type of treatment. And we then repeated that post hoc analysis, including both getting rid of the patients with greater than 15 letters that I already told you about, and then we got rid of patients that had less than 8 degrees visual field. And at that point, we had statistical significance all the way from 3 months all the way out with increasing visual acuity around 15 letters, which is, I mean, absolutely amazing in RP. Now again, post hoc analysis, but I think very validating in terms of planning for our phase 3 pivotal trial. So here's where we are now. We've done quite a few different trials in RP, and in every case, we never saw a case of immune rejection, and we've never had to immunosuppress a patient. So we're here now, poised to enter into the phase 3 trial, after we complete that, we'll file the BLA, which is how you get approval from the FDA for general commercial distribution of the product. Now we call this a neuroprotective strategy. Again, it's gene agnostic, but I think it's also cell agnostic. It can treat both photoreceptors and ganglion cells. It can also treat vascular cells. And so there's a variety of different retinal conditions that might benefit from this very same strategy without even modifying the product. And we have evidence in animal models for the conditions I show here, including optic nerve-related conditions and metabolic, including a diabetic retinopathy model in a rat. So we look forward to extending this kind of work into other conditions. So anyway, thank you very much for your attention. And I just want to acknowledge all the people who've worked on this. And these are only some of the people. It's quite a big effort. And again, thank you for your attention. Yeah.
Okay, thank you. Thank you very much. Very interesting indeed. I would like to know from your experience with the phase 2 trial on the humans, what other than visual acuity parameters did they feel improvement with, and what was studied, and was there any way to test for a progressive degeneration in those who did not improve, or even in those who did improve? Were there any tests done to see if there is progressive atrophy, for instance?
Well, you know, the anatomical changes are slow and gradual, so the follow-up was over a 12-month period, so I think that's somewhat limited. Obviously, functional outcomes, we monitor at different time points, and so some patients improved, some didn't, right? I think, you know, when you think about this as a neuroprotective strategy and you realize there have to be photoreceptors present in the macula in order to improve your visual acuity, you start to understand why some of the patients responded and some didn't. So retrospectively, we can look at our cohort of patients and say some people had a large area of residual macular tissue, central macula. Now, it may not have been functioning optimally, but there was a substrate there that could be acted on by the drug that could return the vision. When people had a really burned out central macula, typically you didn't get much in the way of an improvement that you could measure. Some of these people reported seeing improvements, but I think they were functioning off of more peripheral photoreceptors, and of course they had fewer to work with. So those results are much harder to quantify. In terms of other things we looked at contrast sensitivity, which also responded similarly, but we get this really dramatic effect on visual acuity, so we're kind of going with that. That's a fortuitous result, really. We looked at, there's a visual questionnaire. People differ in their feelings about that, but the mobility part of that questionnaire seemed to be actually pretty valid in terms of these RP patients. If you look at the whole questionnaire, it's generally aimed at people who are better functioning than RP, so it's not really that relevant. We also look at visual fields. That can be harder to test, but we also see similar trends with visual fields. So we think it's not just reading and macular function, but fields in and around the peripheral or the posterior pole.
So this is mostly about preserving the cone function from dying.
It's not just preserving, though, because we're seeing this return of vision, which was kind of exciting and unexpected. So, but you have to have photoreceptors there.
Yeah.
I think the key thing to kind of take away from this is that the photoreceptors are going to get sick before they die. It's a very gradual progression. Now, as ophthalmologists, once the cells got sick and they stopped functioning, we just gave up on them because there was nothing we could do. So even when the cells were sick, they were as good as dead because they were never going to come back. But here now we can distinguish between sick and dead. When they're dead, this treatment isn't going to help. But when they're sick, we can bring them back. And that's why, a subset of people gets a really impressive result here.
Yeah, thank you. Open the question. Floor, yes, please.
Thank you, Dr. Henry, for your excellent presentation. Actually, the challenging question is here, whether these neurotrophic factors that is going to fix the sick cells in RP, whether it's going to really fix it permanently or temporarily. Because as you know, in the future, as any other drugs treating the retina, like the gene therapy, is really cost effective. And we have thousands of patients with retinitis pigmentosa in the region, and maybe the highest number in the Kingdom of Saudi Arabia. So the challenge here of the gel cell, whether, I'm not sure if this is studied or not yet, maybe in the future, because this is just 1 year after the 2B trial. Do you think it's going to fix the sick pathology in those cells permanently, or partially, probably?
I think we already know at least some of the answer to that question, and that is this is not a cure. So what gene therapy hopes to be is a cure, but you would have to do that gene therapy before all the cells have degenerated. So you'd have to go in very early. And if you did that safely and effectively, you could cure it with, one injection. That's not what gene therapy does now, and that's not what we're doing either. So this is a treatment. So it reprograms the metabolism of the cones, and probably the rods as well, and other cells in the retina. But when these cells die, what we see is that the patients slowly start losing vision again. That's why I put so much emphasis on the redosing aspect. So it's a treatment, it's a long-term treatment. It's like an order of magnitude longer than injecting an anti-VEGF. It's not just a monthly thing. It would be more like a year at the minimum. But eventually you'd want to retreat. Now, the economics of this is a whole different thing. Right now, these cells are grown by hand, and so it's a laborious process. So the cost is going to be high compared to something that's manufactured with a machine. But I think as these things are automated going forward, there will be ways to bring costs down long term. But right now, I feel like the key thing is to show that it works, and start helping people. And that creates the momentum that'll allow for us to develop this further.
Thank you so much, Professor Henry. I just want to ask regarding the 160 patients that was written here. How many of them improved to 15 letters in terms of visual acuity?
Let's see. So it's a little more complicated. So 160 was the number of doses. 130 is the number of eyes, I believe, and the number of patients is somewhat over 100. And my colleague here, we're fortunate to have Dr. Adrian Morris, is the person who I would trust to answer that question more than me. So.
The important thing in directly answering your question is, in terms of the total clinical trial program, we've used doses from 0.5 million cells up to 6 million cells. So it's dose dependent. But when we look at the cohort of patients that we're including in our phase 3 program, our expectation is we'll have more than 50% of those individuals who will be improving by 15 letters or more. And the percentage who improve by 10 letters is substantially higher than that. The issue is that the FDA have asked us to set the primary endpoint at 15 letters. To be frank, we personally believe that a 10-letter change in these individuals, a 10-letter improvement from baseline is clinically meaningful. They have told us that they will reserve judgment on that when they see the data. So they've agreed to include 10 letters as secondary endpoint and 15 letters as a primary endpoint. But to directly answer your question, we expect more than 50% of patients will have an improvement of that magnitude. Okay.
Any more questions? I have one last question, if you don't mind. In those patients who have improved, I know that this is considered to be gene agnostic, meaning any RP within certain criteria is eligible to enter. But did you do genetic studies on those that you recruited to see whether there is a tendency for some genetic mutations to behave better than others?
Of course, we do the gene testing on every patient, but you're starting with these small numbers, and it seems like every patient has a different mutation, so it's very difficult to do any kind of analysis. But we will continue to accumulate that data, and I'm hopeful that when we get somewhere around 1,000 patients, we might be able to detect something. But so far, it correlates more with how much macula is around than anything else.
Thank you. Any more questions? We do have another activity, so with this we would like to close, and thank you very much for an interesting lecture.
Thank you.
Thank you very much.